Okay, so yeah, so the cell wall is an essential component of any bacterial cells. And okay, well, what's the link here to say? Yeah, so essential function, all bacteria have got to have them. The central part that is rigid in the bacterial cell wall is a polymer called peptidoglycan, and its synthesis is very, very highly conserved. And this is a kind of weak point in bacteria that can be attacked by molecules that are antibacterial. So lysozyme is going to cleave the polysaccharide part of peptidoglycan, and different families of antibiotics will inhibit the synthesis of the cell wall. Okay, so for example, penicillins, part of the beta-lactams, their function is to inhibit cell wall synthesis. And if this occurs, then the bacteria will lyse. And one of the other things that's very, well, characteristic about the cell wall is that the structure of the cell wall is what differentiates gram-positives and gram-negative bacteria. Now I'm going to go through this now, so everybody is going to do this in the lab class. The protocol of gram staining, probably the most widely used technique in, at least in microbiology lab classes for undergraduates. So the first step in the stain, in the protocol, is to fix the bacteria onto the slide and then stain them all with crystal violet. So this is a basic stain, and it's going to interact with negatively charged molecules inside the bacteria, so RNA, DNA. So all of the cells are going to be purple at this point. The second step is to treat the stained cells with iodine, which is called the mordant. Apparently this is a term from like the dyeing industry. And this is going to form large complexes between the crystal violet, okay, positive charge, and the iodine, negative charge, and make it more difficult to wash out the stain from the cells. Third step is treatment with alcohol. So this is what is going to distinguish the gram-positives from the gram-negatives. So an alcohol wash will remove all the stain from gram-negative cells, whereas gram-positives will retain the stain. Now at this point, gram-positives are visible, gram-negatives are invisible. So to see these cells, you have to counterstain with something else, which is fuchsin, which will give a nice pink color. So what is that telling us? It's telling us that gram-negative cell walls are very sensitive to alcohol, and gram-positive cell walls are relatively resistant. Now, so most eubacteria, not archaebacteria, but eubacteria, can be divided into gram-positives and gram-negatives. And there are some exceptions, okay? So mycobacteria have got a different type of cell wall, not gram-positive, not gram-negative. And there are some bacteria that don't have peptidoglycan at all. And these are the mycoplasmas. Now, as I said at the beginning of this slide, the cell wall is an essential component of the bacterial cell. So how come these guys can live without one? Well, they can live without a cell wall because they are obligate parasites. They're parasitic organisms. They only live on the surface of eukaryotic cells. And so they have a parasitic lifestyle, and they're able to rigidify their plasma membrane by using the same molecule as eukaryotes, which is what? Anybody know? what is preventing the plasma membrane in every cell in your body from just being floppy like olive oil? Well, maybe have some kind of role, but that's not what I'm thinking of. Just a kind of type of lipid fat component that is going to add rigidity to the cell, to the plasma membrane. Cholesterol, yeah. Okay, so cholesterol helps to rigidify the membrane. So bacteria cannot synthesize cholesterol. Only eukaryotes can synthesize cholesterol. But these guys can get away with it because they're parasites. So they will take the cholesterol from the host cell membrane and embed it into their own plasma membrane. So that's how they can have a kind of rigid plasma membrane without a cell wall. But it's an exceptional case. Okay? So for most, the huge majority of bacteria, the cell wall is an essential component. Now, if you look closely at the cell or the wall of gram-positive and gram-negative bacteria by electron microscopy, if you're very lucky, what you can see is something like this. So for gram-positives, this is the cytosol, this is outside the cell, and you can see a plasma membrane and a thick layer here of peptidoglycan. In gram-negatives, what you can see, again, the cytosol here, plasma membrane, a thin layer of peptidoglycan, and an outer membrane. And this explains why the gram-negative cell wall is sensitive to alcohol. Okay? The outer membrane, the inner membrane, these are lipids basically, so they're soluble in alcohol. Just gets washed away. And the peptidoglycan layer is very, very thin. Whereas in gram-positives, the peptidoglycan layer is very, very thick and relatively resistant to alcohol. Relatively resistant, okay? So this is one of the things that can happen in the lab class very often, is during this step, people put the alcohol on, plenty of ethanol, ethanol, ethanol. Everything is going to get washed away, because in the end, if you do this step for too long, then you're also going to wash away all of this peptidoglycan as well. So, it's not 100% resistant to alcohol, but it's more resistant than the thin, highly lipid-based gram-negative cell wall. So that's the basic difference between gram-positive and gram-negative cell walls. Big, thick peptidoglycan layer, no outer membrane. Thin peptidoglycan layer, outer membrane. Now, the common part of the structure here is the peptidoglycan. It's present in both gram-positives and gram-negatives. And it's this peptidoglycan polymer that gives rigidity to the cell wall, okay? Because the plasma membrane and the outer membrane, they're lipid membranes, kind of fluid. Peptidoglycan is what is giving what is conferring rigidity to the structure. So this polymer is, well, it's got a simple function, okay? It's like Kevlar or something like that. It's got to be tough. It's got to be tough. And peptidoglycan, it's got a glycan part, a polysaccharide part, and a peptide part. So the glycan part, the polysaccharide part, is made up of alternating dimers of N-acetylglucosamine linked in beta 1 to 4 linkage to N-acetylmuramic acid. Okay, for us, we're not really interested in this. It's kind of like glycobiology, hardcore biochemistry. The interesting thing, though, is that this beta 1 to 4 linkage, this is specific for bacteria. Now, none of our sugars, polysaccharides have this kind of linkage. So lysozyme, which we can produce, is going to attack this beta 1 to 4 linkage. So it can degrade peptidoglycan, but it won't destroy any of our own polysaccharides. So this, okay, NAM-NAG polymer, it's going to give you a very long linear molecule, okay? So that structure is going to be like this. So you're going to have a lot of like threads hanging around on the outside of the cell. This isn't going to be rigid on its own, okay? So what is bringing the rigidity are these crosslinks, the peptide part. You have 4 amino acids linked to the N-acetylmuramic acid, and these tetrapeptides are linked to each other through a kind of pseudo-peptide bond. Now, something kind of interesting about these amino acids here is if you look at them down here, position 1, L-alanine, position 2, D-glutamate, glutamic acid, 3, diaminopimelic acid in most gram negatives, and 4, D-alanine. So, one of the questions you might be asking yourself is, what on earth is this? Okay, these names might be familiar. Glutamic acid, alanine, right? They're the same as in proteins, right? This one is not an amino acid incorporated into proteins. What's the other unusual thing about these amino acids? Maybe you can spot. What does this mean? Yeah. Yeah, the kind of chirality, the L and the D forms. In proteins, what do you have? What kind of amino acid do you have in proteins? You've got a 50-50 chance. L, yes. So finding D amino acids, that's a bit strange. Okay, so these are 2 clues. This is telling us that however this is made, it's not made on the ribosome, this structure, because you have an unusual amino acid that's not normally in proteins, and you have these D stereoisomers that are never in proteins. So this tetrapeptide is produced in some special way. Okay. All right. So now I want the next thing everybody has to really learn are the major steps in peptidoglycan synthesis. Because well, just because I like it, all right? It's interesting. It's interesting. So why is it interesting? Why is it clever? Okay, because the real problem for bacteria, all right, is they've got to produce and synthesize a big complex polymer outside the cell. So basically in metabolism, when you're breaking something down, this can liberate metabolic energy. Okay, so glycolysis, you break down glucose, you can produce ATP. But if you want to produce something, like synthesize a protein or synthesize DNA, you need to put energy in for the polymerization reactions. Basically because they're condensation, right? So all of these polymerization reactions, you throw out 1 molecule of water. And thermodynamically is very, very unfavorable because water is in a huge excess in the external medium. But that's not the main point here. The main point is, okay, to produce this polymer, you've got to put in metabolic energy. You've got to put in ATP somehow, okay? But all the ATP is on the inside of the cell. And if you were to export ATP outside the cell here, it would be a disaster because it would just all diffuse away. And your bacteria would die of starvation in 5 minutes or something. Okay, so all of the ATP has got to stay inside the cell here, but you've still got to synthesize something complicated on the outside. And this is not a simple problem. It's a very, very difficult problem. And that's why the the biosynthetic pathway is so complicated, because it's, you might just imagine, okay, we'll throw out all the monomers here and we'll make it on the outside. But it can't work like that. It can't work because there's no metabolic energy on the outside. So bacteria have got to do something more complicated. And this is the way it is, okay? So okay, I'm going to summarize the main steps here. So firstly, the repeating subunits. So this is the two sugars, N-acetylmuramic acid and N-acetylglucosamine, complex to a pentapeptide. They're synthesized in the cytosol. So the repeating subunits are put together where you've got the metabolic energy. And then the whole thing is flipped across the plasma membrane by a lipid transporter, which is called bacterial. And then the two, disaccharide type part of the molecule is inserted into the peptidoglycan polymer by a transglycosidase. And then finally, the crosslinks are formed by a transpeptidase. So I'm going to kind of talk you through this YouTube video on this, because it shows all the steps very nicely. If it's going to want to load up. Yeah, maybe. Okay, so this is showing us a kind of diagram of gram-negative cell wall, internal membrane, external membrane, peptidoglycan. And this is a diagram of a gram-positive cell wall, plasma membrane, and a big, thick layer of peptidoglycan. So it's the essential component of the cell wall. So it starts off with uridine diphosphate N-acetylmuramic acid. So what is N-acetylmuramic acid? Okay, so here you've got UDP, so two phosphates here, and this is a kind of like, I'm going to come back here. Okay, so you've got the sugar part here. So this is glucosamine, okay, N-acetylglucosamine here. So there's a kind of N-acetyl part here, and N-acetylmuramic acid is N-acetylglucosamine, but it's got a kind of lactyl group here. So this is 1, 2, 3 carbons here, lactic acid, and you've got a carboxylate group on N-acetylmuramic acid. So this carboxylate group is available to make a peptide bond with the amino acids on the pentapeptide. So that's going to be the first step. Add on the, what is it, L-ala? Let me get this right. Yeah, L-alanine. Okay, so first add on L-alanine. This is the N-terminus of that amino acid. There's a carboxyl terminus. And then it's going to add on the D-glutamic acid here for the peptide bond. So each time these are added, each one of these steps takes 1 ATP to add an amino acid. And then it adds on this weird one, the diaminopimelic acid. And then finally, the D-alanine dimer is added. So you get, well, let's slow down. So you get this N-acetylmuramic acid conjugated to a pentapeptide. And then this is going to be grafted onto bacterial on the cytoplasmic side of the plasma membrane. So this is outside, this is inside. And then N-acetylglucosamine comes on. So you have, okay, this NAM-NAG dimer with the pentapeptide all formed. Okay, this is the repeating subunit is formed inside the bacterial cell. Now it's ready to be flipped around to the other side. Okay, so this is a kind of lipid that can flip from one side of the membrane to the other. So there it is. And now it's going to be, the dimer is going to be inserted into the peptidoglycan. So this is a transglycosidase. So it destroys one bond and forms a new bond. So it's neutral, energetically it's neutral. You're breaking this bond between the phosphate and the N-acetylmuramic acid, so that liberates energy. That's enough to make a new sugar-sugar beta-1 to 4 bond. And then the last step is the transpeptidase activity. So what is exported is a pentapeptide, and the formation of this kind of pseudo-peptide bond is going to liberate the last alanine. Okay, so in the cell cytosol you produce this NAM-NAG pentapeptide, but in the finished structure you've got a tetrapeptide. Now the reason you've got to produce a pentapeptide first is because the energy in that last peptide bond between the two dealanines, this is what's going to allow you to make a new bond. So you break one bond, you make a new bond, it's energetically neutral. So that's why it's so clever, because you've produced this kind of NAM-NAG pentapeptide. It's a repeating subunit, but it's got enough, it's kind of got the energy packed in it to produce a synthesis outside the cell. So it's really good. So what's the last thing to say about this? Okay, so this, the biosynthetic pathway is complicated, but it's highly conserved between gram-positives and gram-negative bacteria. So this is what makes it such a good target for antibiotics, because the biosynthetic pathway is the same in a whole bunch of different bacteria. So the penicillins and the cephalosporins inhibit the transpeptidase step. Vancomycin it actually binds to the D-ala-D-ala dimer, so it's got a different mode of action, but it's inhibiting the same step. The other thing to say about this is that in bacteria which are growing, there's an equilibrium between destruction and formation of the peptidoglycan. Because peptidoglycan is rigid, okay, and if your cell is growing, you're going to need to break some of these beta-1 to 4 linkages, some of these transpeptide linkages, otherwise the cell can never expand, because it will be just trapped in a rigid cage. So in growing bacteria, there's an equilibrium between destruction and formation of peptidoglycan. And all bacteria have got to express enzymes that can destroy their own cell wall. So these are called the autolysins. And that perhaps contributes to the fact that these antibiotics are all more effective on fastly growing bacteria. So bacteria in the kind of a stationary phase can often be a bit more resistant to antibiotics than like fastly growing bacteria. Okay, so we've got about 10 minutes. We're going to be about on time. So last thing about this is the type of peptide linkage is not identical between gram-positives and gram-negatives. Most gram-negatives have diaminopimelic acid in position 3, and the cross-peptide link is direct from the D-alanine of one tetrapeptide to the DAP on the other. Whereas in many gram-positives, the amino acid in position 3 is lysine, and the linkage is not direct, but through a kind of glycine bridge. Okay, that's because the peptidoglycan layer here is very, very thick. So it's useful to form linkages over a bigger distance. whereas in E. coli and other gram-negatives, the peptidoglycan is very thin. Okay, one last thing is that, okay, okay, this is lysine, okay, you've got two amine groups. Which of these two is going to be involved in the formation of this bond here? Same thing with the DAP, okay? You've got two amine groups. Which of these two is involved in this bond? Okay, let's go for lysine. Who says this one is involved here? Nobody. Who says this one? Nobody. Okay, I just don't understand the question. Okay, so well, okay, let's go for the DAP because the bond is direct here. So this is the N terminal here. This is the C terminal, N, C, N, C. So the N, this amine group here, right, this one, that's already involved in the peptide bond to the D-glutamate. Okay, so this one, this amine group is not available. So this is the carboxy terminal of D-alanine, and it's going to be making the link to this amine group here. Okay, it's going to be the same with lysine. That one, this amine group here is down there. It's in this bond, and the interpeptide linkage is with the. It's on the side chain. Okay, the lateral side chain amine group. You have to be clear about that. Okay, so just before we finish, a couple more things about the differences between gram-positive and gram-negative cell walls. So one component that you find in gram-positives but not gram-negatives is teichoic acid and lipoteichoic acid. So these things are polymers of, well, this is a glycerol. Sometimes it's ribitol, so 5 or 3 carbon sugars with phosphate groups. So this is a phosphoglycerol polymer. And R, this can be alanine or glucose or another sugar or another amino acid. So these are long linear polymers with a strong negative charge from the phosphate groups. And their role, so they're going to, and in fact, they'll be covalently attached to the peptidoglycan at one end, and the rest of the time they'll have electrostatic interactions with the peptidoglycan. So these are long linear molecules, and what they're doing is really binding the different layers of peptidoglycan together to make sure they don't come apart. Lipoteic acid is the same kind of thing, but you've got a lipid end group here which is going to attach the whole thing to the membrane. So once again, this is trying to keep the peptidoglycan physically attached to the plasma membrane. In between peptidoglycan and the plasma membrane, this is the periplasmic space. Now in gram-negatives, the cell wall is a little bit more complicated because you have inner membrane and an outer membrane. So because you've got an outer membrane made out of double-layered lipid structure, then this membrane is basically going to be impermeable to a lot of water-soluble molecules like sugars, amino acids. And that's why membrane porins are essential to allow sugars, amino acids to diffuse into the periplasm. So porins are present in gram-negatives but not gram-positive bacteria. Gram-positives, they don't have that problem because this structure is rigid, but it's not impermeable to small soluble molecules. So stuff like glucose, amino acids, they can just diffuse across here. The other thing you find in gram-negative membranes is this thing here called Brown's lipoprotein. So it's got part is in the outer membrane, and in the kind of protein section interacts and binds to peptidoglycan. So this is, again, physically holding the outer membrane, sticking it onto the peptidoglycan, just to prevent the whole thing from separating. So kind of simple structural function. And the last thing we have is LPS, lipopolysaccharide, which makes up the outer layer of this membrane. So you have a kind of polysaccharide, forest of polysaccharide that's on the outer surface of the gram-negative cell. Okay, that's pretty cool. So one more question and then we can break up. So which of the following mechanisms could allow gram-positive bacteria to become resistant to beta-lactam antibiotics? There's more than one correct answer. Everybody got an idea about this? Yeah? Okay. Who says the first one? Express an enzyme that destroys the antibiotic. Who says yeah? Some people. Very frequent mechanism of resistance. Yes, you're right. Beta-lactamases, okay? Mechanism of resistance in gram-positives and gram-negatives. Express a transpeptidase that doesn't bind the antibiotic, so mutate the target. Who says yes? Yeah, that's also a mechanism of resistance. Express a porin that exudes the antibiotic. Who says this one might work? You're right. You didn't fall into the trap. Porins are only in gram-negatives, so it can't work for gram-positives. Evolve a way to survive without the cell wall. Anybody go for this one? Yeah, you're right. It's unlikely. It's never going to happen. Okay. Something else. Yeah. Yeah. Okay. But if you're a spore, you can't grow. The spore can survive, but at some point the antibiotic's got to. We're talking about being able to grow in the presence of the antibiotic. So it's not really the same thing. Something else. I'm trying to think. Yeah, I think these are not the only mechanisms. I think. I think there are other mechanisms in gram-positives, but I don't know them actually. Okay. Maybe I put some in my cheating notes. No, I didn't. I don't have any cheat notes down here. Sorry. So yeah. Okay. So there's more than one way to become resistant. Porins. Okay. This is important for gram-negatives. That can be a mechanism of resistance for gram-negatives. Yeah. And okay, that's about it. So we're on time. Don't forget microbe of the week, 7 p.m. And yeah, anybody have any questions? You have a couple of minutes at the end. Yes.